Antarctica, it's this almost mythical place, right? Pristine, untouched, the last true wilderness.
It really challenges our assumptions about these seemingly pristine environments.
But what if I told you that even there, a complex issue like antimicrobial resistance is rearing its head?
And that's what makes this deep dive so fascinating.
Okay, so for those of us who haven't been to Antarctica recently, can you give us a quick breakdown of what antimicrobial resistance is?
Imagine a constant battle between humans and microbes.
So it's like an arms race, but at a microscopic level.
Exactly. And here's where it gets even more interesting. Scientists have discovered antibiotic-resistant genes in ancient Antarctic ice cores.
Wow, that's mind-blowing.
Some of these genes date back to a time before humans even started using antibiotics.
So even in this incredibly remote environment, resistance to our modern medicines has existed for far longer than we might realize.
It really challenges our understanding of antibiotic resistance purely as a modern phenomenon.
That's what I was wondering about.
These ancient resistance mechanisms might hold clues to how bacteria have always adapted to survive.
Even with limited human presence, we're still impacting this supposedly pristine environment, aren't we?
But of course, we also have to consider the impact of human activity, even in a place like Antarctica.
We are.
And the research shows a clear link between research stations, particularly the larger ones, and higher levels of AMR in the surrounding environment.
That makes sense, but how do scientists differentiate between this human-introduced AMR and what might have occurred naturally over time?
It's a bit like detective work.
So it's like they're piecing together a puzzle to figure out the origins of the resistance, right?
They look for specific clues, the types of bacteria present, the unique antibiotic-resistant genes involved, and even traces of synthetic antibiotics in the environment.
But why is it so important to make this distinction?
Because understanding the source of AMR, whether natural or human-induced, is crucial for developing effective strategies to manage it.
It's like we're bringing our own microscopic battles to a continent that's been fighting its own for millennia.
And this is especially critical in Antarctica, a place we're actively trying to protect.
Speaking of human impact, let's talk about wastewater.
It might not be the most glamorous topic, but it seems to be a significant pathway for AMR to spread even in Antarctica.
It's true, wastewater is often overlooked, but it's a crucial piece of the puzzle.
Think about it. Wastewater from research stations carries traces of medication, including antibiotics, and of course, a whole lot of bacteria.
And even with treatment, some of those antibiotics and resistant bacteria are still making their way into the Antarctic environment.
Unfortunately, yes.
E. coli surviving in the icy waters of Antarctica.
Research has shown that even in these cold conditions, wastewater treatment plants in Antarctica may not fully degrade all antibiotics. One study even found human-derived E. coli, the common gut bacteria, persisting in the waters near research stations.
That's a little unsettling. It makes you wonder what else might be hitching a ride in that wastewater.
You're right to be concerned. We also have to consider things like plastic waste, which is a growing problem globally, and Antarctica is no exception. Studies suggest that plastic waste could act as a raft for microbes, potentially transporting antibiotic-resistant bacteria across long distances.
So it's like a delivery service for AMR, with plastic waste acting as tiny boats for these resistant microbes.
In a way, yes. It highlights how interconnected these issues are: pollution, human activity, and the spread of AMR. Even if these bacteria don't thrive in the open water, they can colonize plastic surfaces, creating a potential reservoir of resistance.
It's a stark reminder that what might seem like a local issue, like wastewater treatment, can have far-reaching consequences. But shifting gears a bit, what about the impact on Antarctic wildlife? Are penguins and seals at risk from all of this?
That's a critical question, and one that researchers are actively investigating. The research is still limited, but there have been some concerning findings. For instance, one study found tetracycline resistance genes, TETM specifically, in Adelie penguins.
Penguins carrying antibiotic-resistant genes. That's heartbreaking, especially considering their image as symbols of pristine wilderness.
It really underscores the far-reaching impact of AMR, and it raises concerns about the potential for these animals to act as reservoirs or even vectors of resistance, potentially spreading it to other species.
That's a chilling thought. It's like this resistance is weaving its way through the entire ecosystem. What about other animals? Have there been any other red flags?
Yes, there have. For example, scientists have discovered human-associated E. coli strains in Whittle seals, which indicates reverse zoonosis.
Reverse zoonosis. So we're essentially introducing a health threat to these animals. That's a sobering thought. What are the potential long-term consequences of this?
That's the million-dollar question. The long-term consequences are still being studied, but it highlights that AMR isn't just a human health issue. It impacts the health of the entire ecosystem. What affects one species can have ripple effects throughout the food web.
It's a complex web of interactions, and we're only just beginning to understand the intricacies. Speaking of complexities, earlier you mentioned King George Island as a focal point for AMR research. What makes this location so significant?
King George Island is home to a high concentration of research stations, making it a prime location to study the impact of human activity on the Antarctic environment. And what researchers are finding there is quite concerning.
Okay, let's dive into that. What have scientists learned from studying AMR on King George Island? Okay, let's dive into that.
Studies on King George Island have revealed some alarming trends.
So we're talking about resistance to some of our most crucial antibiotics showing up in this remote location.
For one, they're finding a high prevalence of resistance to broad-spectrum antibiotics like VOCAMs and aminoglycosides. These are classes of antibiotics that are often used as last resorts for treating serious infections.
That's a worrying sign.
It is. It suggests that these resistant bacteria and their resistance genes are finding their way to even the most isolated corners of the planet. And it raises concerns about the potential for these resistant strains to spread beyond Antarctica.
It's like a stark warning about the global nature of this challenge. If it's happening in Antarctica, it's likely happening everywhere, right?
Exactly. But here's where things get even more interesting. Research on King George Island has also revealed variations in the role of plasmids and mobile genetic elements in AMR propagation.
Okay, can you break that down for me? What are plasmids and mobile genetic elements, and why are they important in this context?
Imagine tiny pieces of DNA separate from a bacterium's main chromosome that can carry genes for antibiotic resistance. These are plasmids, and they can be shared between bacteria almost like trading cards, spreading resistance even among different species.
So it's not just that individual bacteria are becoming resistant, but they're actually sharing these resistance genes with each other. That's a whole other level of complexity.
It is. And understanding how these plasmids and mobile genetic elements contribute to the spread of AMR is a key focus of ongoing research, not just in Antarctica, but globally.
It sounds like there's still so much we don't know about the intricacies of AMR in this environment. And you mentioned earlier that one of the biggest challenges is limited data. Can you elaborate on that?
You're right, the lack of data is a significant hurdle. One issue is inconsistency in research methods. Different studies use different protocols and reporting standards, making it difficult to compare findings and get a comprehensive picture.
It's like everyone's speaking a different scientific language. Having standardized protocols would be like creating a universal translator, making it easier to compare and analyze data from different studies.
A perfect analogy. We also need to move beyond simply culturing bacteria in a lab. Not all bacteria grow easily in artificial environments. Metagenomics, where we analyze all the DNA present in a sample, like a scoop of Antarctic soil, can give us a much broader picture of the microbial diversity and the resistance genes they carry.
So we're essentially expanding our search from just the bacteria we can see to the entire genetic landscape, uncovering hidden resistance we might have missed before.
Precisely. And combining this with whole genome sequencing, which allows us to map the entire genetic code of individual bacteria, gives us an even deeper understanding of AMR mechanisms and how they evolve.
It sounds like we're bringing in the big guns of genetic analysis to crack this case wide open. But it's not just about the science, is it? What about the policy implications? Antarctica, with its unique governance structure, presents both challenges and opportunities in tackling this global challenge, doesn't it?
Absolutely. Antarctica is governed by the Antarctic Treaty System, an agreement signed by multiple nations to preserve the continent for peaceful and scientific purposes. This system could play a crucial role in managing AMR.
So we have an existing framework for international cooperation in Antarctica that could be leveraged to address this issue. That's promising.
It is. The treaty system has already been successful in implementing environmental protections, like regulating waste disposal. We could potentially build on this to enact continent-wide regulations for AMR monitoring and wastewater management.
It's encouraging to know that there are avenues for collective action. Imagine a united front against AMR, with researchers from around the world working together to protect this fragile environment.
And the lessons learned from Antarctica can inform strategies for tackling AMR globally. If we can develop effective strategies to monitor and manage resistance in this remote environment, imagine what we can achieve on a global scale.
This deep dive has been a real eye-opener. I started thinking about AMR as a problem confined to hospitals and healthcare settings, but it's so much bigger than that. It's a global challenge that reaches even the most remote corners of our planet.
And it's a challenge that requires a multifaceted approach, rigorous scientific investigation, innovative technological solutions, and crucially, international collaboration and policy action.
You've given our listeners a lot to ponder today. As we wrap up this Antarctic adventure, what's one thought-provoking question they can take with them?
Here's something to consider. If AMR is indeed a natural process that's been happening for millennia, how do we reconcile our responsibility to address the human-driven aspects of this challenge with the need to preserve the delicate balance of our planet's ecosystems?
That's a question with no easy answers. It challenges us to think critically about our impact on the world around us and to find ways to innovate and collaborate for a healthier future, both for ourselves and for the planet. Thank you for joining us on this journey to the bottom of the world and into the fascinating and often unsettling world of antimicrobial resistance.